Hi everybody, Dr. Ahles here. In this short video, we're going to talk about the process of DNA replication. When we talk about the way that DNA is copied or replicated, we would say that it goes through a process of semi-conservative replication. Remember that semi-conservative replication means that when I make a new copy of that DNA, half of it is my old template strand, and half of it is made out of new nucleotides that I've actually attached to each other. So when we do the process of replicating our DNA, there's two major steps that are involved. Step number one is getting the DNA double helix open. This is going to allow me to read both of the strands of DNA to help me build a copy of them. And after I open up that helix, I need to add new nucleotides all the way along that template until ultimately I've generated two new strands that are half old and half new. So in this process, we'll start with discussing how I open the double helix and then how I add new bases. Step one, opening the helix, involves two main enzymes. The first enzyme for you to be familiar with is called DNA helicase. The job of DNA helicase is to break apart the double helix. Remember that holding each half of our DNA strand together are hydrogen bonds. Remember that between A and T, we have 2 hydrogen bonds, and between G and C, we have 3 hydrogen bonds. So the enzyme DNA helicase works its way through the middle of that DNA strand, breaking apart those hydrogen bonds. As it breaks apart those hydrogen bonds, this starts to put some pressure on places ahead of it in the DNA sequence. So we have an enzyme called topoisomerase, and the job of topoisomerase is to go ahead of the DNA helicase enzyme and make little cuts or small nicks in the DNA. By making these tiny cuts that it'll later go back and fix, this helps to relieve that pressure that comes from unraveling this strand. The best way to think about this pressure is if you've ever tried to pull apart a rope that's made out of several small pieces. As we start to pull those pieces apart, it gets all knotted up until you undo that strain and pull it apart. That's what topoisomerase does, is it gets rid of the places where the DNA would coil on itself and make problems. So, helicase opens up the double helix. Topoisomerase makes sure that that process goes as smooth as unzipping a zipper. When the DNA double helix is open, we end up with 2 different strands, and these strands are oriented different directions. So we have what's called the leading strand that's going to run 5' to 3'. Friendly reminder for us that these numbers give me an idea of what's going on from a chemistry perspective. So we have our phosphate group at one end and our hydroxyl group at the other end. That's my leading strand. My lagging strand is oriented the opposite direction. So 3' where we're opening up the double helix to 5' farther away. This is going to matter because once I start building DNA strands, I'm going to run into some issues with the direction that I can add new bases. So let's talk about how to add those bases. The first thing I have to do to be able to start building new DNA, copying from my template, is I start by making an RNA primer. Now, when I use the word primer, what I mean is just a small piece of RNA. Think of it kind of like a flag showing me where I need to start copying my genetic information. So my enzyme that helps to make this little primer is the enzyme called primase. When primase makes an enzyme, it's important for you to know that what it's built it out of is ribonucleic acid. It's RNA, not deoxyribonucleic acid, which is DNA. The reason that matters is because once I've used this primer to start copying information, I'm going to have to take it out later. So primase adds RNA to specific locations on the template strand. That RNA primer is going to allow DNA polymerase 3 to actually start putting DNA nucleotides onto my copied strand. So DNA polymerase 3 looks for that flag, looks for that primer. When it finds that primer, it attaches that location and starts to move from 5' to 3', adding bases as it goes. Remember, the only side of a DNA strand that I can add new bases to is the 3' end. So we'll always see DNA polymerase 3 traveling in a direction that looks like it's going from 5' to 3'. As it's doing this, it can attach new bases to that 3' end of what it's building. Now remember, with our DNA double helix, that when I go through and open it up, I make this thing called a replication fork. So the replication fork is just the two sides of my original DNA molecule. As they open up and as that moves along that double helix, we see two new strands of DNA that are being made. Now we've talked about those names. We've talked about the idea of having a leading strand and a lagging strand that are made. Now we're going to talk a little more specifically about what's going on with those two strands. So let's start with what's called the leading strand. When we talk about the leading strand at the replication fork, we're talking about my strand of DNA that's being built in the direction toward the replication fork. So DNA polymerase 3 is copying this base, and then it copies this one and this one and this one. It's moving the same direction as that replication fork is moving. Now, that's good news for us because that means that the 3' end, where I can add bases, is moving closer to that fork. Ultimately, we make one continuous strand of DNA with that leading strand. That's perfect. That set of genetic information is good to go. It's a little bit harder for me though with the lagging strand, because when I talk about the lagging strand, the direction that I'm adding new nucleotides is actually opposite of the direction that the replication fork is going. So the 3' end, where I would be adding bases, is going away from the replication fork. As the fork opens, that's the 5' end of these fragments. So what that means is the lagging strand is actually made in several small pieces. The name of these pieces are Okazaki fragments. And I want you to underline, highlight, star Okazaki fragments. I make Okazaki fragments, or these small pieces of DNA, when I'm building my lagging strand. Because my lagging strand, the direction that I add bases to, is actually going away from that replication fork, meaning I'm going to have to start over and over and over every time this fork moves that direction. So, big idea to remember. The leading strand gets built in one piece, nice and easy. The lagging strand built in many small pieces because of the direction that I can add those bases. When we're talking about that lagging strand, the strand that makes the Okazaki fragments, each Okazaki fragment is going to have its own RNA primer. So notice I put a primer on right here in green, and I started to build a DNA copy off of it. Excellent. But then my replication fork moves over to the left. Now I've got to put a new primer on and add some more bases. And when this moves to the left again, I'm going to have to add another RNA primer. So the lagging strand is constantly adding new RNA primers to keep restarting the process of building a strand. Meanwhile, my leading strand has one primer, and we just keep continuously adding bases to it as we go. So here's the enzymes we've talked about so far. We talked about DNA polymerase 3. The job of DNA polymerase 3 is to add nucleotides after that RNA primer. Once we've added our bases to that RNA primer, and the enzyme called DNA polymerase 1 comes back and checks the work. RNA polymerase 1 has the ability to cut out those RNA primers and replace them with DNA nucleotides. Because remember, RNA is ribonucleic acid. I'm trying to build deoxyribonucleic acid. So DNA polymerase 1 will get rid of any of that RNA that's in my new DNA molecule. But once I've replaced those primers with nucleotides, there's still one more task I need to do. What I need to do is connect these Okazaki fragments that are now entirely DNA to each other. And my enzyme that does that is DNA ligase. So DNA ligase comes in at the very end and acts kind of like glue, ligating or grouping together all of those Okazaki fragments to end up with one continuous strand, the same kind of strand that I made with my leading strand, because I got to build that in one piece. So overall, let's look at this DNA replication picture. Remember that DNA replication happens first with us opening the double helix. My enzyme helicase helps me to do that. Once I open up the double helix and have two halves that are opened up, I can then start building my DNA. Remember that the leading strand is the strand of DNA that I can build continuously. So on my parent strand, we're going from 5' to 3' away from that fork, which means when I go to build my strand, I'm going 5' to 3' into the fork. This strand made continuously. But because the lagging strand is oriented backwards, because we're going the other direction with that strand, remember that we make that in small pieces. So we've talked about all of the enzymes that we see here on this picture. Helicase, opening up the double helix. Primase, adding those RNA primers. DNA polymerase 3, which adds my sets of DNA bases. DNA polymerase 1, that gets rid of the RNA primers. And finally, DNA ligase, which helps me to piece together all of my Okazaki fragments. If you want all of that in one place, here's a great summary table for you that lists all of the enzymes that we talked about and their job in replication. Now, I should mention that when we copy our DNA, our cells are pretty good about proofreading their work. So DNA polymerase 3 by itself is able to look at the sequence of the template strand and the sequence of what we just made to decide if something got added in incorrectly. If DNA polymerase 3 realizes something incorrect was added, it'll actually remove that base and try again. Try to put the correct base pair in there. So every time your DNA is replicated, not only do we build that sequence, but we also check it as we go. And the good news too is if DNA polymerase 3 happens to forget or happens to look over one of those incorrect bases, we have what are called mismatch repair proteins that'll go in that place and fix that for us later. We also have what are known as nucleotide excision enzymes. Nucleotide excision enzymes work on a little bit bigger of a scale than our mismatch repair proteins. These enzymes actually go in and will cut out sections of your DNA that has been damaged. So for example, exposure to UV radiation can lead to areas where our thymine bases that are next door neighbors to each other, they'll just connect to each other instead of across the DNA double helix. That would be something that a nucleotide excision enzyme can come in. It can cut out or excise that piece of DNA that's broken, and then DNA polymerase 3 can come back in and add new bases. So as we're thinking about the process of replicating your DNA, know all of our enzymes that are involved, and know that we do also have some proofreading mechanisms present in the cell.